You're listening to Antiviral Frontiers with your host, Rick Kiesit. In this episode, we're joined by medicinal chemist Dr. Derek Lowe, renowned for his blog, In the Pipeline, as he shares his extensive drug discovery experience in the pharmaceutical industry.
Welcome to Antiviral Frontiers. Yeah, please introduce yourself.
Hi, well, I'm Derek Lowe, and by trade, I do drug discovery. I started off as an organic chemist, then I turned into a medicinal chemist. Now I'm more of a chemical biologist, and I've worked at several different biopharma companies over the years, but I'm also well known for a blog that I have written, for over 23 years now, called In the Pipeline, which talks about chemistry, drug discovery, and a lot of other scientific topics.
Awesome. Yeah, I've looked at your blog. It's incredible. You've been at it so long. What have you learned anything really interesting from that process of blogging for that long?
Oh, definitely, yeah. It's been good because it has forced me to keep up with the literature, and anyone who has a scientific field knows what that's like. It's like sticking your head in a fire hose stream. But it has forced me to keep up with the literature, and it's also forced me to learn about a lot of topics that, honestly, I probably would not have learned about. And, of course, I've met a lot of people I wouldn't have met otherwise, so it's really been good.
Yeah, sounds incredible. It could be interesting. So what we're working on here at ChimeraMed is a family of broad-spectrum antiviral therapeutics. Ours are large molecule compounds, and our audience is generally fairly technical.
Okay.
It would be, I think, interesting if you could walk us through your experience and impression of the drug development process. I think a lot of people don't really have their arms around that very well.
Oh, it's true. That's one of the things I've learned over the years in writing about drug discovery and development. And in fact, that's one of the reasons I started the blog, and I started writing about it because when I would tell people at, a party or whatever, what I did for a living, they'd say, "Oh, that's really interesting. I didn't know that was a job." So
right.
That's how I decided, people will find this interesting and no one seems to know about it. So why don't I talk about it? So yeah, it has changed over the years because I'm now in my 36th year of actually working in the pharmaceutical industry. That explains the 4 different companies by now. And it has changed a bit over the years, but the general outline of it is the same. There are really only 2 ways to get started on a drug discovery project, and this applies to antivirals as well as every other therapeutic area. You can have a target-focused approach where you have a rationale based on what you know about the biochemistry of disease, or you can go in with a phenotypic screen where you set up an assay that you believe in your heart of hearts recapitulates the disease state, and then you just start throwing compounds at it to see if you can find something that makes that better. And those are still the 2 main ways to do it.
Right.
But once you do that, in either way, you're going to get a bunch of compounds that had some kind of activity, either against your preferred target, which was probably running an assay against an isolated protein and the like, or you got a bunch of compounds that affected your system, your cells or small animal model in the way that you wanted. Now comes the hit to lead process, because all of those were hits in your assay, but believe me, they're not all going to be leads for drug discovery and development. A fair number of them are garbage, but you don't know which ones. And it is your job as a hit to lead person to winnow through this as quickly and effectively as possible. So one thing you'll do is you'll go through and look at what other assays these compounds might have hit in. And you might get a little worried if it turns out they have hit over and over and over in all kinds of assays against all kinds of targets. That is what we disparagingly refer to as a frequent hitter. And it's not impossible for one of those to turn into a real drug, but the odds are stacked against it. Meanwhile, there may be some on there that have never hit anything else. That might be really neat, or it might be the sign of an artifact, that this was just an accident and this thing actually didn't hit in your assay either.
How many compounds are you typically working with?
Yeah, the
these scenarios?
screen, we don't screen on the scale that we used to back in the 90s. Big companies would screen hundreds of thousands or even low millions of compounds. We're a little more focused now, but even so, you'll screen tens or hundreds of thousands of compounds without really batting an eye if you have the compounds and can afford to do it. Hit rate, of course, will determine how many hits you get out of the assay. And there are some targets and enzymes, for example, kinase enzymes, tend to have a pretty high hit rate, especially when you screen them against collections from drug companies, because we've done an awful lot of kinase work over the years, so we're probably going to have some kinase hits in there. Other things like transcription factors, oh boy, hit rate there might be 0. It just might. And if your hit rate turns out to be something like 3 or 4 or 5% on a transcription factor, that means your assay was wrong.
Right.
Something screwed up. You do not honestly have a 5% hit rate on a transcription factor. That's never happened. But at any rate, at the end of the process, you're going to have, depending on your cutoff, at least a few dozen compounds that you really feel like you need to investigate.
Okay.
So you look at those and you see what they've hit before. You look to see if there are any other compounds similar to them in your compound libraries, and you get those tested. You also check to see if there are things like this that you can buy, that look similar. We call that analog by catalog, and you test those. The idea is that you're looking for an SAR, a structure-activity relationship. Are there recognizable changes in activity as you do small changes in structure to the compound? If there are, you might be in business. That's what we can work with. If there aren't, you may be dealing with a singleton, a compound that works, appears to be real, but every single thing you do to it makes it worse. Well, that doesn't give you much room to maneuver, so you're generally going to drop those. And you're going to work with series that show that they have some legs, some SAR that you can actually work on. This made it a little better, this made it a little worse, et cetera, et cetera. And you pick those probably to prioritize.
So would you call that lead optimization?
Exactly. That is where you switch gears to lead op. And it depends on how many chemists you have, how many different series you can take into lead op. Sometimes just one. Sometimes 2, 3, or 4. It depends on just how luxurious life is for you at that moment. But you will start making analogs around these things. And you'll do that based on what the early SAR told you. But in many cases, especially at the beginning, you'll just make analogs to start covering some chemical space. We don't have anything where we mess with this end of the molecule. Let's mess with it. We don't have anything where we flipped this chiral center. Let's flip it. We don't have anything where we change this oxygen to a nitrogen. Can we do that? And you just try to spray a bunch of interesting stuff into the chemistry space and see what happens. Most of these will not work. Most of everything doesn't work. That's a painful lesson that especially chemists have to learn when they join drug discovery and development, because they've often come from very high-powered organic synthesis groups, and you have these 32-step syntheses of some incredibly weird natural product, and you're troubleshooting the retro-Zamboni reaction at step 22, and you're not going to be doing that in the drug industry. You start a 36-step synthesis, you're going to be fired.
Right.
You've got to do things and make analogs as quickly and cleanly and as interesting a chemical space as you can. So a 5% yield that gets in a vial for the assay tomorrow is infinitely preferable to a 95% yield 6 months from now.
Oh, yeah.
So much preferable. And that's a lesson that you have to internalize.
Yeah, sure. It makes sense.
Oh, yeah. So you crank out as many of these as you can and you start to get the landscape. Okay, this target or this activity, if it's a phenotypic assay, seems to like it when we make this end of the molecule more polar or less polar. Or this end over here likes it when there's a hydrogen bond acceptor, et cetera, et cetera, et cetera. And you start to develop some hypotheses about how your compound might be binding. Now, at the same time, you are obviously trying to do computational modeling if you have the protein target that you're looking for. If that target can be crystallized, then you are definitely trying to crystallize your ligands in this and get things like X-ray structures. So you want all the structural information you can get, and you want the modeling people to see if they can get their teeth into it and come up with some hypotheses. But really, nothing beats experimental data. Now,
Is this like basically an evolving method of action here as you go? Is that what's happening?
And you have to be ready to discard your hypotheses along the way because just as in most of those compounds are probably not going to work, most of your ideas about the compounds themselves are probably wrong. Right. And you can't get too attached to them. That's another lesson that, of course, any scientist has to learn is that, the universe is under no obligation to make you feel better about how smart you are.
Yeah, very true.
Oh, yeah. So you have to take it as it comes, data rule. And you have to be able to trust your assays and the folks who are running the assays. If you don't, well, you're kind of hosed at the very beginning. Yeah. But if you do, and you all know what you're talking about, and these are folks you've worked with, and you know how things are going, then, odds are you can make some progress. If we have a series with some real SAR in it, one way or another, we can often hammer it into shape. And by that, I mean make them potent enough, selective enough, long-lasting enough under physiological conditions like the bloodstream, well absorbed enough where they'll get out of the gut if you take them orally as a pill, and so on and so on. Generally, we can come up with something that will get to where we want, or at least pretty close.
How much does formulation come into play in this stage?
Oh, yeah.
In your experience?
In the early stages, not much, because these things are not even seeing animals. You really get into the formulation talk when you start narrowing down on one or two series, or even just two or three compounds that might be the winners. And this is when you're really starting to pick clinical candidates. And at that point, you've probably been putting compounds into whatever small animal assay you have. But then you really start to get serious about, all right, what's the tox? 2-week tox starts to come up if you're talking about, a real clinical candidate. 2-week tox in at least 2 species. Generally, small animal, large animal, rat and dog in the classic combo.
Okay.
So you're going to have to be able to make enough of this compound to do that, because in tox, you're going to be dosing it up to fairly high levels. And honestly, you want to see something happen when you do that. It's nerve-wracking when you dose a compound up really high and it still doesn't show any toxic effects, because you're like, well, I know you're out there.
Yeah.
So, but you need to have enough compound on hand to do that. And at this point, of course, you are talking formulation, because you have to be keeping a close eye on the actual blood levels and pharmacokinetics. What's the area under the curve of this thing? What's the half-life? Is this sufficient for once-a-day dosing? Do my AUCs and half-lives match up between my small animal and my large animal? Can we use that data to do allometric scaling and figure out what it might be if we go on to a human, and so on? So formulation really does start to play a role there. You don't want to play too big a role. Ideally, you just want to have something where just a good old-fashioned, garden variety formulation will work just fine, but that's not always the case.
Have you found good correlations between your in vivo models and humans? I know this is an issue for us. We face
Oh,
working
yes.
with infectious disease, and a lot of the animal models are really pretty terrible.
Well, it's interesting you should say that because I have worked in a lot of different therapeutic areas. I've done antivirals, I've done antibacterials,
Okay.
I've done CNS, schizophrenia,
Hmm.
Alzheimer's, I've done metabolic disease, diabetes and obesity, osteoporosis, a lot of different kinds of oncology. And honestly, when I look back over the whole area, one of the areas that I think has absolutely the best and most reliable animal models are infectious disease.
Okay.
That you may find that kind of terrifying because we may both be right. The other one, by the way, is
Yeah.
diabetes. The animal models
Yeah.
for diabetes are pretty good.
Yeah.
But the animal models for, say, central nervous system disorders are absolutely horrible.
Mm.
Just, just terrible. The animal models for the CNS stuff are just notoriously unreliable. And I've learned that several times in the early part of my career, which actually made me resolve never to do CNS again.
Right.
No Alzheimer's, no schizophrenia, no depression, because we don't understand these things at all. And the animal models are
Yeah.
not very predictive. Another area
Yeah.
that's very tough is immune function,
Mm.
because human immune systems are so different. Oncology is kind of in the middle because you can do xenografts with human-derived cancer tissue into immune-compromised mice. And if
Mm.
your compounds don't work in a rodent xenograft model, they're probably not going to work in humans. But if your
Yeah.
compounds do work in a rodent xenograft model, they're probably not going to work in humans either. So, as I
Have,
say, it's kind of in the middle.
have you had a chance to work with humanized mice,
Yeah,
with
yeah,
humanized immune
it's an
systems?
improvement. But of
Yeah.
course, humanized mice are graded on a curve. There's a whole spectrum of just how humanized are these. And since, especially for tox, we don't know a lot of the tox mechanisms very well. We know the ones to look for. We look for signs of liver damage through, enzyme elevations, ALT, AST, phosphatase, whatever. We know to look out for compounds that hit the hERG ion channel because that tends to be correlated with later cardiovascular problems, torsade de pointe, and other things. But most of the rest of it is just a horrible thing that drops on you out of a clear blue sky.
Hmm.
So we don't have any animals that are humanized enough to warn you about human tox.
Yeah. Okay. So you come out of lead up and you go into preclinical, I guess, would
Yeah,
be
yeah. At this point,
next. And
you start putting things into these 2 week talks and everything. You try to see what
hmm.
makes it through. Generally, something will, not always. I've been on projects where nothing made it through, and we went back around,
Mm.
tried to figure out what went wrong and how to fix it. Or we even notched back further and went back to one of the earlier chemical series if we believed in this target enough and went back and tried to develop something new from that. That takes some real grit and some real perseverance on the part of upper management, but it can happen. But
Is
in
it usually a
general,
tox issue that'll bounce you out of
yeah,
preclinical?
yeah, because if you put it
Mm.
in, yeah, it usually is because you don't often have an animal model that's so beautiful on efficacy that it will totally kill you. Although, if you didn't have one you at least somewhat trusted, you probably shouldn't have started the project anyway. This
Yeah.
is what really torpedoes a bunch of phenotypic projects, and I've seen this happen. A company will start off a phenotypic project in a really tough area like neurodegeneration or something like that, and they'll have this really whiz-bang neuronal culture thing going where they think they can determine phenotypic changes. And the chemists and biologists will labor away on it. They'll find several different series because in phenotypic assays you usually find things that do 3 or 4 or 5 different interesting phenotypes, and which one's most relevant to disease? Yeah, you got me.
Mm.
So you'll end up with this surplus of riches. And at that point, you don't have an animal model that you trust well enough to go into humans. You're standing on the side of this huge canyon, and most companies tend to balk when they say, well, we got this stuff that works great in our phenotypic model. Let's go to people. And they're like, you must be joking. You've got to have a transitional model. And you're like, we didn't have one when you started. Did you think we were going to come up with one in the last 18 months? And so that can be a problem. But yeah, generally your compounds, if you're serious about them, do work in whatever efficacy models you have come up with and you've agreed among yourselves to believe it's tox that'll kill you. Or maybe it's PK. Maybe they work, but you can't get them at high enough blood levels or long enough blood levels to be a real once-a-day compound. Yeah, something like that,
Or PD. Does PD ever bite you?
Oh, excuse me?
Does PD, pharmacodynamics,
Yeah,
distribution?
Sure. It can, especially with something like CNS. You're like, okay, we have great big levels in the periphery and it's not getting into the brain. And at that point, some of your less experienced biologists will turn to you and say, well, why don't you guys put that thing on there that makes it get into the brain? And you ask them, what thing is that exactly?
Right.
But you will have some things also where compounds will ride around on plasma protein forever, and you'll just have this weird volume of distribution. That I'm not too scared about plasma protein binding. There are people who get really nervous about that. I'm fine with it. It's an equilibrium, and the way I see it is just a nice controlled release. But you do have some cases like those bisphosphonate drugs for osteoporosis where the stuff rides around for, 4, 5, 6 months. And they decided that was a feature, not a bug, and that's how often you take those drugs. But you do have to be mindful of this sort of thing while you're developing.
Yeah. So you finish preclinical, and then if that's successful, you move on to
it's
an
about
IND.
Successful as I've ever been. Now I have to say the last 10 or 12 years I've spent a lot of it in very early stage chem bio research. So I'm not putting a lot of shots on goal into the clinic. But even in the earlier part of my career, I never, ever had anything get to phase 2.
Okay.
And that is not just because I'm a hack. I might be a hack, but it's not just because of that. It's because, as many of your listeners will know, the failure rate across clinical trials in this business is about 85% overall disease indications.
Yeah.
Vaccines are by far the best. The failure rate in vaccines is only about 50%. It's beautiful. The failure rate in CNS is a nice solid 95%.
Wow.
Oh, yeah.
And they fail mostly in phase 1 or later?
Unfortunately, no. Yeah, that's the problem. Compounds used to fail a little bit more often in phase 1, but phase 1 these days is mostly about making sure that your pharmacokinetics estimates were right and that you've got the appropriate dosage. And we've gotten better at that. I have had a project that wiped out there, and we had to go back and fix that. Mostly, though, you get past phase 1. Now, if tox shows up in phase 1, that's a showstopper for sure, and everyone goes home. But you get into phase 2. Most of the failures are in phase 2 for efficacy, which means you pick the wrong target. It doesn't do what it thought you did, or it doesn't do it enough. Or they're in phase 3, and the phase 3 failures, that's where you start to get these horrible tox out of nowhere as you start going into more and more patients and a wider variety of them for a longer period and at different doses. It's like, oh crap, what happened? So that's where the failures are. So we managed to spend the most amount of money we can possibly spend before we find out that things are bad.
So for our listeners, maybe you could explain the difference between phase 2 and 3.
Oh, sure. Okay, phase one, as I say, is just going into normal human volunteers, or as normal as you can be in volunteer to take an investigational drug, but you're going into people without the disease and just checking blood levels and gross safety. Phase two is going in for the first time to people who actually have the condition you're trying to treat, but you're going in very carefully, generally a smaller number of people, enough to be statistically convincing, or you're wasting your time, and some people do, but you want to go into patients that you think will give you the best chances of success. So you pick them very carefully, and it's almost like a little greenhouse where this beautiful plant is growing, and there are people who carry that too far. And then when they get to phase three, which is where you branch out and you start going into a larger number of patients, more diverse patient population, more reflective of the real world that you would be selling this stuff to, maybe you made your phase two just a little bit too sheltered and perfect, and it wipes out there. But phase three is the thing that's supposed to be, all right, we've shown that it seems to be safe. We've shown that it actually does seem to do what we thought. Now let's see if it can actually be a drug. If we give it to the young, the old, the people who are taking other drugs, people who have different levels of severity of disease, and so on and so on. And that's
Right.
phase three. So they're larger and more expensive. Every step of the process is more expensive than the one that came before.
Yeah. And there's nominally a phase four in theory, right?
Yeah. And that varies depending on what therapeutic area you're in. But yeah. And of course, many times these days you can get some sort of conditional approval from the
Sure.
FDA, and they will require you to do post-marketing studies. I think the FDA may have gone a little bit too far in that direction, because there's an awful lot of post-marketing studies out there that were supposed to have reported by now and haven't quite gotten around to doing it real soon now. Just you wait. We'll keep selling the drug while we do this, of course, but we're on it.
Yeah.
So I'm a little peeved about that.
As a medicinal chemist, I'm curious if you've at what stage in the development cycle you address or run into issues related to GMP fabrication.
Oh yeah. That starts to kick in when you're really picking a clinical candidate. Because before that, in the discovery stage, there's really no GMP, GLP stuff going on at all. You're just banging these compounds out any way you can.
Yeah.
But when you start to get serious about something that might go into humans, that is where the regulatory paperwork starts. And it just increases along the way. So you, of course, are going to have to take all of that 2-week tox data and all your PK studies and all that. You're going to have to take that to the FDA to get an IND. Start with, just to let them take you into humans at all. So it has to be up to standard. I don't even do that kind of work. That's in a totally different lab with totally different people, chemists and everyone else. I have never done GLP type work. I would suck at it.
Yeah, I understand there's also lots of material specifications, limitations, restrictions, and so on, animal free or,
Oh, yeah, absolutely.
Yeah.
And just the chemistry is a different world. That's the world of the process development chemists, where they're trying to get chemical routes that are extremely reproducible and especially produce the same impurity profiles every time within a narrow range, so you don't get weirdo surprises based on some impurity that started popping up. So they're in a very demanding position. I respect those folks a lot, because they're having to come up with chemistry that can be reproduced, can be done on scale, can be done with starting materials that you're always going to be able to buy in quantity and whose purity is going to be the same every time, that doesn't have a terrible waste disposal problem, that doesn't need exotic conditions that's going to cost a lot to turn into a manufacturing process, you're really playing a very difficult puzzle game there.
Also, the quantity production
Oh yeah.
requirements are significant, right?
Oh
You
yeah.
have to be able to make quite a bit of it. It's not the same as,
You do.
flask scale or
Oh yeah.
whatever.
Totally different world because I mean, I do a lot of my stuff on a few milliliters of solvent. And it
Right?
doesn't really matter what you run a 2 or 3 milliliter reaction in. It can be a nice little round bottom flask. It can be a scintillation vial. It can probably be, a container that used to have breath mints in it. I mean, I haven't tried that, but it could be almost anything
Yeah.
because that's a very benign environment. But as you start scaling up, you run into problems, say, with mixing and heat transfer. If your reaction gives off some heat, you are going to have to figure out where that goes. On a large scale, that can cause terrible problems, as in overpressures and exotherms and, 500-liter flasks pouring out all over the floor, and nobody wants that stuff.
Yeah, that's for sure.
Oh boy.
And so I gather most of your work has been with small molecules. Is that
It
correct?
is. It is. I have really never been a biologics person, even though now I am more of a chemical biologist. But
Yeah.
I'm on the small molecule end of chemical biology. As I say, I trained as a synthetic chemist. I was one of these people
Yeah.
working. I had a 27-step synthesis of a natural product antibiotic, rosarimycin, and I have never worked on anything that lengthy again. Thank God.
Do you have some sense of what the differences are or likely to be on the biologics large
Oh,
molecule side?
certainly, certainly. It's, of course, a different world because you are there, more likely than not, hijacking all of these wonderful enzymatic and biological systems that evolution has honed up for you, starting with PCR and working your way up from there. And it is a different world. I mean, we are just Cro-Magnons over in chemistry by comparison. We're saying, me put compound in flask, heat up very hot. Me react compound. Whereas, over in a more molecular biology oriented area, it's like, all right, we are going to take this beautiful enzymatic system that evolution has optimized, and we're going to fool it into doing our work for us. And some of these things are just beautiful, starting again with PCR. But that goes to all sorts of other things, all the kinds of enzymatic systems you use to make, modified cell lines to produce your proteins or to serve as assay models, all that sort of stuff that we can do. And of course, we now have all these things like CRISPR and the like, single base editing. It's just incredible.
Yeah. Yeah, it's an amazing and wonderful world. It's advancing so quickly even today.
It is.
Yeah.
It is. It's terrifying. And where ChemBio comes in is where you try to marry that to organic chemistry. A simple example would be an antibody drug conjugate. Antibodies, of course, are amazing. You have trillions of them running around your bloodstream, all different. And they are privileged proteins. Your body does not destroy them like it does most other proteins. They can wander around there for months and years. So it's very tempting to modify these for drugs. But at that point, you're having to do some organic chemistry on them because you're actually reaching in with small molecules that might be the payload and forming covalent chemical bonds with bridging groups that stick onto the protein sequence of the antibody. So how do you do that? Well, you should call a synthetic organic chemist.
Right. Yeah, very interesting. What are your views on IP in the drug development space?
Yeah. We, of course, guard it very jealously. And it is pretty much what makes drug discovery in the modern Western world run. is the expectation that you will have some patent protection. And that's because of that 85% failure rate. Almost everything that seems bizarre or just enraging or peculiar, inexplicable about the drug industry, generally it traces back to the fact that we have an 85% failure rate. No other industry does. 85% of Pizza Hut's pizzas are going to be as good as they're ever going to be. 85% of Toyota's cars can actually roll down the road without bursting into flame or exploding. Right. 85% of our stuff blows up and takes all the money with it. So that means when we find something that actually works, we have to squeeze the maximum amount of money out of it because we never know when we're going to get another one. I worked for a company that ended up getting bought by someone else because they were getting so much of their revenue from one drug that went off patent, and there wasn't anything good enough to replace it. And they mishandled the patent expiration pretty grievously. The stock lost like 70% of its value, and someone scooped it up.
Terrible.
Yeah.
Wow.
So IP is a serious business, but it also seriously pisses off a lot of people. And I understand why. When I write about it on the blog, I can see the readership statistics dropping almost in real time. Those are invariably the least read of whatever I've written for a month.
Wow.
Yeah, it's like,
have you noticed any, I guess you could call it trends in the direction of using, say, trade secrets instead of patents or in addition to patents?
Yeah.
Given that trade secrets don't expire and patents do?
Right. Yeah, that's a good point. There have always been trade secrets over in that process chemistry area. And that's there to confound the generic drug makers when they move in when the patent expires. Because there are some little tiny tricks of the trade about, oh, you actually want to use this other solvent. You know, or you actually don't want the temperature of that reaction to go above 43 C. We have found that out, but we didn't quite put that in the patent. Of course, patents require you to disclose best mode.
Yeah.
Or they can be invalidated. So it's always you're walking a tightrope there between what you're patenting and what you're going to keep as a trade secret. The other problem is, of course, if someone figures out your trade secret, you have no recourse.
Yeah.
So, oh, well, now they know how to do it too. So I still see the trade secrets as mostly being in these weird little recondite tricks in the drug scale-up business. And not so much for the earlier stuff, because the earlier stuff, a lot of the IP is composition of matter.
Sure.
What is the exact structure of this lead compound, and what are the structures of the other compounds you're claiming around it? And that pretty much, as far as I can see, has to be explicit. Yeah.
I know one thing an issue that we have as a biotech in the patent space is just the whole, well, first of all, it's only a 20-year, 20 years sounds like a long time. It's not really that long, given a 10-year development plan or program. And then you have the PCT issue with the international patents, and so you get 30 months, I think it is.
Yeah.
And then after that, you now have to pay an annual fee, like per country it's licensed.
Exactly. So, as you nationalize these things. Yeah.
I mean, can you talk to that? Have you run into this sort of thing?
Oh yeah, it's a major pain, and I think there are people who start small companies who don't quite realize what they're letting themselves in for there. It's a significant amount of effort and eventually expense to keep all that up. And you really have to have people who are paid just to do that. And that is why you have a legal department, focusing on IP issues, because it's not something you can do part-time.
Yeah, indeed. And so for a biotech like us, let's say,
sure,
who has who's made it through lead op and through preclinical and through phase one, and now a business model that I've seen, and it's of interest to us here actually, is out licensing
Right, right.
drugs. Do you have some impression about what it is that pharma companies are looking for when it comes to in licensing
Money.
on their end? It's like there seems to be a trend that I've noticed that some companies have actually, like some pharma companies, have apparently closed down their research departments entirely, and they're switching entirely to
If you know of
in
anyone who's
licensing
done
mode.
that, I would love to hear who they are. I'm not aware of anyone who's actually done that. I've heard stories about this for the past 30 years,
Yeah.
and I think that there are some large companies, and I would suspect Pfizer is one of them, that have run the numbers on exactly doing that and have decided not to.
Okay.
And my feeling on that is that a couple of reasons. One is that you need to keep this sort of expertise in-house so you can evaluate the deals that people are bringing you.
Mm.
And you also need a plausible backstop. If you say, well, look, we don't have to deal with you guys. We could do this ourselves, you know. Well, no, you can't if you don't have an R&D department anymore. And I think that means that if a company were to actually announce, we just ditched all our R&D. We are now your licensing shop for regulatory affairs and approval and marketing. Come on down. I think
Yeah.
if some company actually did that, the price of all their deals would go straight up
Mm.
because now everyone knows you have to make a deal.
Right.
In fact, you have to make quite a few, and there are not that many wonderful deals out there to be made. And I'm not sure if at any given time there are enough of them to float a good-sized company
Mm-hmm.
on the basis of just those deals, because, of course, there are other companies out there who might want to make a deal too.
Yeah.
And they all know that you have to because you don't have anything else other than deal making. So this is why I have not seen anyone. Well, I believe it was Allergan for a while that was trying to do this, and they crashed and burned with it. I've had to look at that. I think that's who it was. But to the best of my knowledge, everyone who has tried to do that has eventually come to grief over it. And they've mostly been small scale players. No really large pharma company has ever announced that. Now, the shareholders would probably cheer deliriously if they did, because God knows the R&D departments are just a endless black hole with red ink pouring into it. And it would immediately improve your bottom line. But after a while, I think the disadvantages would become apparent. So for small companies, I think in-licensing is almost invariably where they end up going. Because what's your alternative? Develop it yourself. Ho-ho. Which means raise an awful lot more money and hire a bunch of people who do the things that you don't do and haven't done before and don't know how to do yet. So you can do it. Small companies have taken the whole thing through themselves, but what that generally means is they end up firing everyone who was in R&D along the way. And this happens again and again and again. It's happening right down the street from me in Cambridge as we speak. I'm not going to go into who, but you can probably pick more than one, because they're like, okay, we got to get this thing through. Sometimes it's not even, they're not even going to take it to the market. We just have to get this thing through phase 2 before we can interest a big company in a licensing deal. We don't have enough money to do that if we still have an R&D department. Sorry, guys.
What do you think the big companies are looking for in those sorts of licensing deals if they're trying, negotiating or talking with biotechs about
they're looking for easy
licensing
money.
their drugs?
Of course, they're looking for easy money. I have that sounds rather glib, but it's true. I have a rule that says almost any question that can be formulated as, I wonder how come the answer is money.
Okay.
And try that out. You'd be surprised how often that works instantly. But that's what they're looking for. They are looking for I'll be a little more elegant about it. They're looking for a favorable risk-reward profile, as in they want something where you take all the risk and they reap all the reward. The
I see.
traditional method of negotiating with small companies on the part of a big one has been, so you guys need a deal real bad? Here's a really bad deal. What do you think?
Yeah.
And again, this sounds cynical, but I hate to say it, but there's a lot of truth in that. The large companies are looking for something that you have de-risked as much as possible already and has good commercial prospects. And you don't have the money to realize those, and the big company does. Or the big company has the expertise to get it through the more advanced trials that you don't have the money to do. And that's generally how it's split up. But if you come to them with an incomplete story, as in, well, we think this is going to do X and Y and Z, they'll be like, come back when you know.
Yeah.
We have a bunch of stuff already that we think is going to do something.
Right. Yeah.
So it's tough. It's not an easy business. It's not an easy business if you're a big company trying to get your own drugs through and look for additions and bolt-ons and deals to make. It's even tougher if you're a smaller company where your entire future might be tied up in this one program. There are small companies that remind me of some of the early ramjet type designs where they would drop the experimental, unmanned plane off of a larger plane at high altitude and let it head down toward the ground and pick up speed so it could ignite its engines. And if it
Right.
ignited the engines, it could pull up out of the dive. And if it didn't, well,
Yeah. I'm curious if you've encountered much, I don't know, I guess, friction or resistance with regard to the way that big pharma is engaging with academia of late, like let's say over the last maybe 10 years, something like
it's
that.
actually gotten better over my career.
Okay.
That may be another
Has it?
terrifying thought in the same way that we had one about the animal models. It's
Yeah.
actually gotten a little better because there used to be a lot more distrust and outright animosity because the old guard of academic chemists and biologists tended to feel that real scientists, really good scientists, stayed in academia. That
I
was the
see.
highest thing that you could aspire to, was to get yourself the best possible position at the best possible university and have the biggest possible group with the most possible funding. And if you didn't do that, there was something wrong with you. After all, that's how they did it. And that's how their
Right.
advisors, their famous advisors did it, right? This is the
Right.
whole academic family tree. So that meant that people who went off to work in industry, well, you know, the type, they weren't as good or they sold out. If you thought they were good, well, you were disappointed in them because they didn't do what they really should have done and become famous academic professors. No, they went off and worked for money, Now,
Yeah.
there is a lot of bullshit in that attitude. And there's bullshit in the attitude of industry toward academia too. Because on the industry side, after being burned a few times in academic collaborations, you'd have people who get really cynical and go, yeah, these folks think that they have absolutely got 3 Nobel Prizes waiting for them. Their ideas are solid gold. They will refuse to entertain any kind of realistic offer because they've got the most epochal discovery that anyone's ever seen, and we're just too stupid to appreciate that. I mean, there are some academic institutes that will come in and start naming figures right off the top that will just make you start laughing.
Mm.
And this has actually improved, though. One of the reasons has been that there is more collaboration between industry and academia, and because of all the turmoil in both areas over the years. There's been more cross-pollination. There are more people in academia who used to be in industry. Now, there have always been a lot of people in industry who used to be in academia. Every single one of us started out as grad students and postdocs, but there are also people who have left faculty positions and decide, the heck with this. I'm going to start my own company, or I'm going to go join a big drug company. So that's actually helped bridge the gap some. So as nasty as the relations sometimes seem to be, I think they're at an all-time high. You can be a glass half empty or half full person about that statement.
Yeah, that's good to hear. What's your perspective on the near-term future of drug development? Particularly with the advent of AI and all that other, there's a lot happening, I think, in addition to that. I see it mostly on the large molecule side, but maybe it's happening on small molecule too. I don't know.
I think that's true. And I think the reason you see more of an AI impact on the large molecule side is because we're talking mostly about protein sequences there. And that is something that AI/ML, and honestly, most of the time when people say AI, they mean machine learning, but that's something where that can really make a difference, as witness AlphaFold and RosettaFold. Now, these programs are not godlike oracles. They will choke if you give them a protein that has weird old folds in it they hadn't been trained on. But otherwise, they're really pretty damn good. Startlingly good in their way. So if you're working in a constrained language space like that, we have 20-odd letters for the amino acids. We have 4 for the nucleotide bases. If you're working in a constrained language like that, you can actually learn a lot. Plus, we have huge amounts of data to feed them on things that have worked. And if you want to generate things that didn't work, and you need that for ML, you can generate a lot of those pretty easily too. So it's actually a very good situation for AI ML approaches. For small molecules, not so much. And the problem is there's a ton of hype in this area where a lot of people in the press, and maybe even in some companies, think that, this is going to increase our rate of drug discovery. We're going to choose our targets better. We're going to be better this way and that way and the other way. We don't know enough to de-risk targets. We don't know enough to feed all this stuff into an AI ML model and predict tox. Nowhere near. So all the companies that are doing computational and AI machine learning methods, once they take a compound up to clinical development, they have now joined the same race as all the rest of us. All they did was get to the buzzsaw a little bit faster.
Right. Yeah. Interesting. I'm also curious about your impressions or views on what might be like particularly underappreciated aspects of drug development.
You mean underappreciated by the general public or underappreciated by... Okay. The general public does not realize how little we know about what it is we're doing. Think about it. Think of all the things we get surprised by. Think of the big discoveries that get made where we didn't... how many types of RNA were there when you were in grad school compared to how many types of RNA are there now? Yeah. And what are they all doing? Well, we don't actually know. Are there more of them? Yeah, probably. Etc. So there's just so much that we don't know and don't understand. So I think the AI stuff is actually making this worse because people are getting the idea that, this is a solved problem. We'll just ask, the wonderful, amazing general intelligences where the next drug is coming from and how to treat diseases. You even hear stuff like this coming from a lot of people who damn well should know better.
Yeah.
But overall, I think that in general, people don't realize how in the dark we are about things. And that's, again, why we have an 85% failure rate.
Right. Yeah. Very interesting. Do you have any tips for biotech companies like ours on things we either should be doing or shouldn't be doing in our drug development work?
It's tough, though, because every situation is a little bit different.
Sure.
You of course, I think this is not, as Shakespeare says, it needs no ghost come from the grave to tell us this, but I think that everyone should be working on things that are going to be actionable. That is, you have a defined disease, you have a defined problem, and you especially have a defined patient population, so that when you find something, you know how you're going to set up your clinical trial, you know who you want to treat, and you know where to find them and how. That can be tricky sometimes, but you need to have that in mind. You have to have an elevator pitch. Can you explain in 15 seconds why your company exists and what it's trying to do? If it takes you 10 minutes, you need to refine that, because there's got to be a way to get it down.
Yeah.
In the same way that I tell people you need to have that ready in case someone appears at your door at a big company and says, "Why do you work here? Why do we pay you?" So you
Yeah.
need to have a good answer for that, but you need to have a good answer for why your company exists. What are we doing? What are we doing that's different from everybody else? And do we have a reason to believe that we have an edge? Are any other people trying to do what we're doing? If not, why not? Do we have some really hot knowledge they don't have? Well, great. Just don't fool yourself. Are
Yeah.
lots of other people trying to do what we're doing? Well, why are we better than they are? Because having a flow chart that says, "and then we get lucky," is not a very good business plan.
Yeah, that's for sure. Well, thank you so much, Derek, for your time. It's been great chatting with you. Can you let our listeners know where they can find you online?
Absolutely. I blog at Science Magazine, so that's science.org, and I believe it's just science.org /blog/pipeline. But if you just search for the phrase "in the pipeline" and start to put in the name Derek Lowe, it'll come up right up there. You'll get a few hits related to a guy who used to pitch for the Boston Red Sox, but I have been gradually eclipsing him as he's been retired from baseball for longer and longer.
Okay. Excellent. Thank you so much.
We hope you enjoyed this episode of Antiviral Frontiers. To hear more from the fascinating world of viruses, biotech, and medicine, please visit our website or find us on Spotify and YouTube and follow us to get future episodes.